Okay, that sounds good. Connie, you can start the recording and we're ready to go. Go ahead, Paris.
My name is Paris Bazemore, and my project is called Isolation and Identification of Plastic Degrading Bacteria from Environmental Water Samples in Zobius Mario. We depend on plastics in our everyday lives. We need it to store our leftovers in the fridge or to carry around water in plastic water bottles. However, have you ever thought about what happens to our plastic waste once we're done using it? Our plastic waste breaks down into something called microplastics. These microplastics can be ingested by animals such as birds, shrimp, and fish, which can kill them. These microplastics can also travel up the food chain and find their way into animals we eat, such as fish, and these microplastics can cause oxidative stress, DNA damage, and inflammation in humans. These microplastics also take hundreds of years to break down. However, a bacteria known as Idonella speeds up this process. It's able to break down PET, one of the most commonly used plastics, and it's frequently used to make water bottles. And it can break down PET into BHET, also known as bis hydroxyl ethyl terephthalate, and BHET into TPA, also known as terephthalic acid. PET and BHET are not biodegradable, but TPA is. Normally, to break down PET, it takes over 450 years. However, Idonella is able to speed up this process. Additionally, Zobius mario is known to eat plastic. It's a species of darkling beetle, and its larvae have been observed to eat polystyrene and polyethylene, which are two different types of plastics. Changes in the gut microbiota have been observed in Zobius mario after the larvae are fed plastic, and its ability to eat plastic is inhibited when it's fed antibiotics. This indicates that the bacteria within the gut of Zobius mario are likely responsible for its ability to eat plastic. However, the bacteria responsible for this ability are unknown. The objective of this study was to collect and isolate bacteria from water samples in the intestinal tract of Zobius mario, and then to test the bacteria's ability to use PET and BHET as a carbon source, and then to identify the isolated bacteria using its 16S ribosomal RNA sequence. My hypothesis is that the bacteria within the intestinal tract of Zobius mario and water can use BHET or PET as a carbon source and thereby degrade microplastics in the environment. The first step in the study was to collect the water samples. To do this, I went around local lakes and ponds and streams and collected water samples. But for Zobius mario, I needed to feed the larvae 1.5 grams of polystyrene for 2 weeks. After this feeding period was over, I dissected the larvae and extracted its tract, and its intestinal tract was then placed in 5 milliliters of distilled water, and the solution was then vortexed. Once the samples were collected, 50 microliters of each sample was pipetted and spread onto an M9 BHET plate, and 20 microliters of each sample was pipetted into a PET 1X M9 minimal salt solution. This solution provides bacteria with all of the nutrients they need to grow, but it doesn't provide a carbon source, which is provided as the PET pellet suspended in the solution. Similarly, with M9 BHET plates, these plates provide bacteria with all of the nutrients they need to grow, except for a source of carbon, which is provided in the BHET suspended within the plate. After these samples were pipetted onto a BHET plate, the plates were then left to incubate, and I searched for zones of clearance on the plate. Zones of clearance are areas of transparency on BHET plates where the agar is more transparent due to the BHET suspended in the agar being degraded by the bacteria growing on the plate. And here to the left, you can see an example of a zone of clearance. It's labeled A and B, And on these two areas, I used a toothpick and I rubbed the toothpick against the zones of clearance, which are labeled A and B on this plate. And then I rub this toothpick onto an M9 glucose plate. All bacteria are able to use glucose as a source of carbon. So once these bacteria were spread onto an M9 glucose plate, they were able to grow much more quickly. And you can see that on this plate there are a variety of bacteria growing. You can see small pink colonies growing as well as small yellow ones. After the bacteria were first streaked onto an M9 glucose plate, they were then isolated based off of morphology, their size, shape, and color. And then PCR was used to amplify its 16S sequence for identification. The 16S sequence is a highly variable region within bacteria that's able to determine or let you know what bacteria it is because it varies so much between different types of bacteria. After the bacteria were isolated, they were then tested on M9 BHET plates, and signs of degradation were then recorded in PET tubes. Degradation was recorded in PET tubes based off of the decrease in concentration of PET pellets suspended in the solution, as well as signs of bacterial growth. Bacterial growth was noticed in these tubes based off of cloudiness in the solution, which you can see with your eye. Here, PB2 is an example of a bacteria that could use PET as a carbon source, And here you can see that the tube is very cloudy and that the pellets seen earlier in CB2 are not visible in this tube, which indicates that the bacteria growing inside of it is able to break down PET. The isolated bacteria were also streaked onto M9 no carbon plates. This was an important step because autotrophic bacteria are able to use CO2 in the atmosphere as a source of carbon. So these bacteria are able to grow on M9 BHET plates as well as M9 no carbon plates. This was problematic for this experiment because with these bacteria, I wouldn't know if they're using the BHET and the M9 BHET plates as a source of carbon or CO2 in the atmosphere as a source of carbon. So if a bacteria grew on both M9 BHET plate and a M9 no carbon plate, testing with this bacteria was discontinued. Of all of the bacteria isolated, 10 of them could use BHET as a source of carbon, 3 could use PET as a source of carbon, and 31 couldn't use either. The number of days it took for the bacteria to grow on a BHET plate varied. Here you can see that DGPHB11 required 5 days of incubation before the first visible colony was noticed. However, in DGPHB1, it took 20 days before the first visible colony grew on the plate. PHB is a substance bacteria produce when they have excess energy. It's similar to glucose, and you can see that for all 3 of the bacteria, they both grew on PHB plates for 5 days before the first visible colony was noticed. However, the amount of time required for the first visible colony on a BHT plate varied. And here you can see the phylogenetic tree I created using the 16S sequences of the bacteria I isolated. And I'd like to draw your attention to the phylogenetic tree because in it you can see that a variety of bacteria were isolated. There were Streptomyces, Verovorax, and Burkholderia. And of these, so there were a variety of bacteria isolated in this study, but of those that could degrade plastic, most of them were Streptomyces and Rhizococcus. This surprised me because I thought that it would be just one type of bacteria that could really degrade plastic because I thought the ability was really rare, but it turns out that it's more than just one type with the ability to use BHET and PET as a source of carbon. A total of 41 bacteria were isolated in the study. 10 of the isolated bacteria could degrade BHET. 3 of
2
the
minutes.
isolated
You have 2 minutes.
Okay. 3 of the isolated bacteria could degrade PET, and 3 bacteria isolated from the intestinal tract of Zilbius mario could degrade BHET. Bacteria isolated in the study could potentially be used to remove microplastics from the water and the environment, and the bacteria isolated were not previously known to degrade BHET or PET. In the future, I would like to sequence the genomes of the isolated bacteria capable of degrading PET and BHET so I can get a better understanding of the genes associated with its ability to degrade plastic. Also, more research is required to gain a better understanding of the degradation pathway the bacteria were using to degrade PET and BHET. Thank you.
Nice presentation, Paris. Very